Hey Beaklings, today we're going to talk about complex inheritance patterns. So you should feel very comfortable with Mendelian genetics at this point. You should understand complete dominance, right? Big T, little t is still tall, right? Little t, homozygous, true breeding short, things like that. You should understand 1 and 2 gene crosses and how to set them up using Punnett squares. You should understand that Punnett squares are really just showing you probabilities, right? The rule of multiplication and the rule of addition. You should understand those. You should know things like test crosses and pedigrees, okay? So phenotypes, genotypes, all of that has to be in your head already. But if you're feeling good about all that stuff, then you're ready for today. All right, so let's take a look here and get into some complex inheritance patterns. All right, here we go. So what we're going to talk about here is going beyond Mendel. Okay, so for example, Mendelian patterns are great and they always hold true for the most part, but once you start looking especially at like human genes and things like that, oh boy, you get way more complex. Okay, but a lot of them are simple Mendel, and that's why Mendel was able to crack genetics, because he kept it simple and he kept his experiments very controlled. Okay, so when recessive alleles are not completely dominant or recessive, oh, that changes things, right? Everything we've talked about so far was like big T, little t is still tall. Well, what if big T wasn't completely tall or completely dominant? That'll change things. You can see we could have multiple alleles today. So far, everything we've been talking about was like big P for purple, little p for white. That's it. What if there was big P, little p, Z? All right, that's going to change things up. And then we have multiple phenotypes, right? Everything we've talked about so far is just 2 phenotypes. But what if there was more than 2 phenotypes, right? The physical expression of those genes. So it's going to get a little complex today. All right. So let's see here. We have complete dominance is what we've been talking about so far. It occurs when phenotypes of the heterozygote and dominant homozygote are identical, right? So that's what we were saying again, complete dominance, big T, little t, and big T, you can't tell them apart phenotypically. You look at those 2 pea plants, they're both tall, right? So that's complete dominance. But 2 major curveballs that we're going to talk about today are incomplete dominance and codominance. So incomplete dominance, that's when the phenotype of the F1 hybrids are somewhere in between. Okay, now that's the definition of incomplete dominance, but here's the way I like to think of it. When you have true breeding dominant and true breeding recessive, okay, which is saying the same thing, and then you have a heterozygote. So think of the 2 homozygotes, and then think of the heterozygote. If that heterozygote is a blend, okay, which is what people used to think, right, the blended hypothesis, if that heterozygote is a blend, well, then you're looking at incomplete dominant. Okay, this is not true, but imagine if it was like the tallness gene. So that would be big T is tall, little t is short, big T, little t, medium. Okay, that would be incompletely dominant, but you know that's not the case. Big T, little t is still tall, so that's completely dominant. So we'll take a look at incomplete dominance. And then codominance means you're both expressed, right? So think about it. For example, big T is tall, little t is short. What would big T, little t be then? Tall, short? How could you be that, right? So codominance is literally when both alleles are completely expressed. So that one's a little confusing, and we'll get into that in a second here too. Okay, let's take a look. All right, let's start with incomplete dominance here. We have snapdragons, okay, and snapdragons exhibit incomplete dominance. And some of the times I think like, ooh, thank goodness Mendel didn't study these and he studied pea plants instead, because if he did, he might have gotten his F1 here and saw they were a blend and thought, oh, okay, we're good, right? Because that's what people thought would happen. But Mendel didn't. He studied completely dominant. Let's see what happens here. So you have this red flower. Okay, so we're looking at one gene here, and the gene is for flower color. So as you can see, this is homozygous red, RR. And you could write it still like big R if you wanted to. It doesn't matter. They're using this nomenclature here, so that's fine. So C big R, okay? And then for color gene. And then over here you have CW for white. Okay, and like if I was doing this problem, I might have picked like little r here. Okay? And what's happening is you see this plant, well, he's a true breeder. He has no chance to give anything except for red, the red allele to his kid. And this one over here has no chance except to give the white allele to its kid. But then you can see here in the F1, we're pink. And that's brand new, right? For the first time since we started genetics together, we have 3 phenotypes. And the middle phenotype, the heterozygote, is a blend of both homozygotes. Red, red, white, white, you put them together, pink. That's incomplete, right? As soon as you see the heterozygote is a mix of the 2 homozygotes, like a blend, then you know you're talking incomplete dominance. Okay? And then what's even crazier, so imagine if you're Gregor Mendel, he might have crossed these 2 plants, gotten pink, and said, well, I'm finished my work here, right? Everybody was right, it's a blend. But maybe not, because look what happens when you mate the F1. Now if we mate pink and pink, okay, and if you look down here, here's that cross. We have pink, pink, and look what you get. 25% red, 50% pink, and 25% white, right? So incompletely dominant, it's easy stuff. It's just a curveball. I like to call these things genetic curveballs today, because you're so good at Mendel now, I hope. And now here we go, perturbing the system and let you know, like, sometimes it actually changes from that. All right, let's take a look at another example here, which is a disease called hypercholesterolemia. Okay, and I actually have a friend who has this. He's a very healthy guy, and he's a runner and things like that, but he actually, when he was like 40 years old, he had a bypass surgery because he built up too much cholesterol in his arteries, and he actually lost his dad before he even had a chance to meet him. His brother died at a very young age, so this is a genetic disorder in his family. And what it is, well, remember, DNA tells RNA to tell the ribosome how to make a protein. So all of us, hopefully watching this video, have the DNA, right? That'll get translated into this protein right here. And what these are LDL receptors. LDL is called low-density lipoprotein. Okay, so these guys are filled with a lot of these cholesterols. All right, and there's new research all the time about cholesterol and different things, but pretty much we understand that the LDLs are bad for you. HDLs are high-density lipoproteins. They're a little better. Apparently, they can clean out some of the junk in there, but LDLs are like little sticky droplets that can get stuck places. Considered, like maybe we talked about it with lipids earlier this year, like maybe in your coronary arteries around your heart, right? And that causes atherosclerosis, or that can cause an occlusion, right, and seal off those blood vessels. So this is very dangerous. But what do most of us do? Well, as this LDL floats around in our blood, our cells have receptors, and once this guy touches here, our cell will take this in through endocytosis. And if you look here, this person has a mild disease, and look at his genotype, or her. Big H, little h has some receptors. And now let's go over here. Severe disease. Okay, this poor person here received DNA from both parents, and neither parent's sequence of nucleotides was correct for telling the RNA to tell the ribosome how to make LDL receptors. Right? So hopefully all of us watching are this. Okay, hopefully none of us are this. And there are some people out there that are big H, little h. Do you see why this is incomplete? Right? Remember the rules for incomplete dominance. It's you have both homozygous conditions. Big H, you're good. Little h, very diseased. But big H, little h, you're a blend, right? And that's why this is an example of incomplete dominance. Okay, let's go on to our next one. All right, so here we have codominance. And codominance is an interesting curveball because this inheritance pattern, when you have a heterozygote, both alleles might be completely expressed, okay? And generally they are completely expressed for codominant, but we're going to come back to blood a little later and talk about how it has 3 alleles. But anyway, you can see here some common ABO blood groups, and this is a table that talks about how common they are on the planet, but as you can tell, they're different depending on what your ancestry is, right? Because don't forget, humans inhabited all over this planet, but then it wasn't until recently that we really started jumping around and things like that. So you can tell genetic patterns based on what your ancestors were because they became best fit for whatever environment on the planet they lived in. So for example, here you can see that in Caucasians, the rarest blood type, okay, and this is, I guess, across the board pretty much here, is AB negative. Okay, you might be saying, Mr. Boylan, what's your blood type? And I say, please, of course it's A+. That's all I do, A+ work, baby. All right, here we go. Let's take another look at codominance. Well, this, what are we doing here? That's not appropriate. Why am I talking about Guinness? Well, that's my buddy. That's my dog. And if you knew my dog Guinness, now this was a couple dogs ago at this point, I brought him to the vet because, oh, look at that tongue. And I thought, wait, what's going on with his tongue there? And again, he was super, super black, right? Was his coat, and that's why we named him Guinness. But he was such a good dog. He was so cute. He actually, we used to have a teacher here that taught physical science, and he came over to my house to play poker once, and my dog, it was like a cartoon, he bit right into his butt. He pulled his back pocket off. I have no idea. He wasn't a mean dog, but he didn't like that guy. Anyway, so what is it going on here? We took him to the vet, and I said, oh, look at these black spots all over his tongue here. And the vet said, well, you probably don't understand this, but there's this thing called codominance. And I thought, oh, okay, I'm a bio teacher. And I said, oh, well, you know what probably happened? It's probably your dog is a mutt made from a black lab, because he really looked like a fluffy black lab. And they said probably a chow. And chows have these like purple tongues, whereas black labs have pink tongues. And they said on his tongue there, they're both expressed, right? It's codominance. It's not like it's a pinkish purple or anything like that. And it's not like it's just purple or just pink. It's both at the same time, and that's codominance, right? Incomplete is when it's a blend. Complete is when only the dominant shows up. Codominance is when they both show up. Okay? So my old buddy Guinness is a good example of codominance. All right. So there's something else about blood here that we have to talk about, and that is multiple alleles. Okay? So this is kind of like another weird curveball where most genes aren't simple like what Mendel studied. Most genes don't just have a big T and a little t and things like that. Most genes are very complex and have multiple alleles, right? If you're bored, look up bunny rabbits. Bunny rabbit fur, it's really cool. There's Himalayan, chinchilla, albino, all these different cool furs. So that's multiple alleles. So most genes have more than 2 possible alleles, right? And imagine the phenotypes then. So, for example, let's take a look at blood. The 4 phenotypes of the ABO blood group, they're determined by 3 alleles for an enzyme that attaches the A or the B carbohydrates. Okay, so remember on the outside of your plasma membrane, a lot of times you have these carb chains, and I said they act like chemical, like ID cards. Okay, well, some of your carb chains on these blood cells are you can consider them antigens because your immune system can read them. But anyway, these carb chains are A's or B's, okay? So, and it's named for this enzyme that attaches them. So actually, if you look here, this is generally, if I was talking about the A allele, normally for blood, you write it like a capital I with an A superscript. B would be this, okay? Would be the capital I with a B superscript. So what is this little I right here? That's showing that you do not have any of these carbohydrate ID cards on that blood. You're missing it. So what's the genetic condition? Oh, so I don't have DNA to tell my RNA, to tell my ribosome how to make that enzyme that puts the carb chains on my blood. And what is that type of blood then if you're missing the A or the B? O. Okay? So if you have no A or no B, you're type O. Okay? So the enzyme coded or encoded by the IA allele as the A carb, and the enzyme that codes for IB puts the B carb on there. Let's take a look here. Okay, so if you have IA, okay, this allele, you're going to put, this is what they're making the carbohydrate look like in this book. Okay, so let's take a look down here. If you're AA, that means you're type A blood. Okay, and again, we're not talking about positive or negatives right now. We're just talking about the A, B, or O. So right there, that rhesus factor, though, the positive negative thing, that's just a different carb that's on your blood cells, okay? And if you're positive, you have it. If you're negative, you don't have it. Okay, so anyway, this is A, but take a look over here. Oh, if one parent gave you type A and the other parent gave you O, you're still just A, type A. What does that tell us? That tells you that A is dominant over O, right? Completely dominant, right? Because look, AO is still A. So A is completely dominant over O. All right, let's take a look at B. So B, they're going to look like these little circle carbohydrate antigen things, right? So BB, if both parents gave you B, well, of course, you're going to be type B. Now, what if one parent gave you B and the other parent gave you O, where you don't put any carbs on there? You're just type B. So we just learned something else. B is completely dominant over O, because BO, right? That's fine. BO is just type B. Okay? Now, the next one is interesting. Hopefully you could tell me what this is. What if one parent gave you A and another parent gave you B? Look at this blood. It doesn't have just A or just B. It has both AB. Now, what pattern of inheritance is that? When the heterozygote, both are completely expressed, codominant. So AB blood is actually an example of codominance, right? All right. And if you take a look at our last one here, what if both parents gave you O? Right? Little I, little I. Well, then you don't make the enzyme that puts those carbs on there, so you're just type O. Okay? And see how this is multiple allelic? Instead of just having 2 alleles for the ABO blood group gene, you have 3. You have the A allele, you have the B allele, or you have the O allele. Right? And because of that, you could have AA or AO, that's type A, BB or BO, that's type B, AB or BA, right? That's type AB, and then OO is type O. So A and B are completely dominant to O, but A and B are codominant when together. Pretty cool, right? So that's multiple alleles. Now there's some cool stuff with blood groups here. Let's talk about who can give blood to who. Right? And I know we here at Pennsbury, hopefully you guys get involved with the blood drive when it goes on. They do so much good, right? And they get so much blood out of you guys. They suck it out of you. But you have to be a certain age and a certain weight and all that kind of thing. But it's a really good thing if you can get involved in that. Good job. Saving lives. So if your blood group O, we already talked about what your genotype is. That means both parents had to give you type A. But here's the thing. Antibodies are these little proteins that your white blood cells put out to protect us, right? So think about if you're type O, and now this is the blood that's being mixed in with your blood. So let's say you're getting a transfusion. If you get type O from somebody, your body says, okay, that's fine. So now you have type O blood floating around. But if anybody gave you A or B or AB, well, what's going to happen is you're type O. And all of a sudden, your type O blood, don't forget, has nothing on it. It's just these smooth little donuts without holes. These squishy little donuts. They float around in your vasculature all day. They carry oxygen on their hemoglobin, right? Blood cells are really just like little baggies. They don't even keep their DNA once they're mature. And they only last, like a third of a year or something like that, because they're always going through valves and getting punched in capillaries and things like that. So, I mean, you make more in your big bones, actually, in your marrow. That's called hematopoiesis, when you make more blood cells. Anyway, if I have type O blood and somebody put A inside of me, or B or AB, my immune cells are going to say, wait a minute, what are these carbs that I've never seen before? Kill it, kill it. And they're going to start trying to attack that blood because it's not me. They don't know who that is. And that's why the pictures here, they're trying to show that it'll clot, right? The blood will coagulate, right? And you do not want a clot traveling around in your vasculature. Okay, that's very dangerous. It's an embolus, right? If you have type A blood, okay, that means that one parent gave you AA, one parent could have given you AO too, or one parent gave you A, the other parent gave you A, one parent gave you A, the other parent gave you O. You're still making type A blood. So up in the O, we would kill anything A or B. We make anti-A or anti-B antibodies. Well, if I'm type A, take a look what happens when I give O. It's okay. Do you know why? Think about that. If I'm type A right now, which I am, if somebody put type O blood inside of me, okay, my immune cells would say, there's nothing to feel. There are no carbs. So my immune system cells would say, go ahead. I guess you're good because I don't see anything out of order. Even though I should have A, there's nothing there. So if somebody gives me O blood, I'm still okay. And if somebody gives me A blood, my immune cells would say, oh, I feel you A. But that's okay because I'm A. So it would let it go. But if anybody gave me B or AB, take a look at the chart. Oh, right? Now it's going to coagulate. It's going to clump up, and that's very dangerous. Okay, so I would make anti-B antibodies if I'm type A. Now let's take a look at type B, right? If I'm type B, each parent either gave me type B or one parent gave me B, one parent gave me O. It doesn't matter. I'm still type B. Now I'm going to make anti-A antibodies for the same reason, right? But look again, if you gave me O, my white blood cells would say, I think you're good, right? There's nothing to feel, so O goes along. If you give me B and I'm type B, no problem. But if you gave me A or AB, my immune cells are going to realize it, and they're going to kill it, and it's going to clot, right? Okay, and let's take a look at the last one here. So what if you're codominant, right? What if you had AB? Okay, guess what? Look at this. You don't make any antibodies. You're not attacking anything. And hopefully you know why, right? Think about it. If you're sitting there right now with AB blood, if somebody gives you O, your immune cells say, well, I don't even know what's going on there. That's fine. If somebody puts A in you, it says, oh yeah, A, I remember you. That's like my A on my AB blood. If somebody gave you B, same thing. No problem. And if somebody gave you AB, hey, I know both of you antigens on there. Go ahead, you're fine. Right? So, if you're AB, you're in good shape. Okay, so you can take blood from anybody. You know what they call that? Okay, AB blood is the universal recipient. So if you have AB blood, doesn't matter. Now again, the positive and negative does matter, okay? But for right now, we're not really talking about that. And what do you think type O is? Look at the chart there. O can give to anybody. So O is called the universal donor, right? So if you happen to be O negative, right, and you go and give blood, well, that's good because the Red Cross will call you constantly, right? They're always going to want your blood. And that's a nice thing, right? That's the gift. And there's all these cool things coming out anymore anyway with blood, with synthetic bloods and things like that now that we can use. So blood is getting really, really, really cool. But that's a little bit about blood right there. And don't forget, what did we cover with blood now? Well, we covered that it could be complete dominance, like A is dominant over O or B is dominant over O. It could be codominance because A and B could exist together at the same time. And the reason that we talked about blood here was because it has multiple alleles, right? It's not just A and B, it could also be O. So it doesn't always end with like purple, white flowers. It could be purple, white, green, or yellow. Oh boy, right? So multiple alleles could exist for, and do exist for lots of genes. Okay, let's move along here. Okay, here we have this phenomenon that happens with genetics. It's pleiotropy, okay? Or some people say pleiotropy. So in pleiotropy, most genes have multiple phenotypic effects, okay? So this again is going to go kind of right in the face of what Mendel was studying. For example, if he's studying flower color, you know what that gene did? Determine the color of the flower petals. How tallness, how tall is the plant, right? Things like that. In pleiotropy, you're talking about when one gene, sure, it has maybe even a simple genetic system, like here we're going to talk about sickle cell, okay, which is kind of like an incompletely dominant on a cellular level because the sickle, if you're a carrier for it, your intermediate blood cells are a little sickled, okay, so I guess you could think of it that way. It's a recessive disease. If you have sickle, that'll kill you. Now, we talked about the Anopheles mosquito causes malaria, so actually it's okay and it's good to be a carrier, be a heterozygote, okay? But in this case, we're not talking about the genetic inheritance pattern. We're talking about pleiotropy. And pleiotropy is when one gene causes a whole bunch of effects. Okay? And that's a little confusing here. So, as you can see, and lots of genes do this. Okay? So here you can see sickle cell, and you might say to yourself, yeah, I know what sickle cell is. That's when red blood cells are sickle-shaped. Yes. Okay, but what does that cause then in the body? Don't forget, you're a system. Okay, you're a system of a human being. So just because you have one gene that's a little different maybe than somebody else, that doesn't mean that that's where the story ends. That can perturb or affect your system in different ways. So in this case, these red blood cells, look what happens here. Well, first of all, you have anemia, right? Because they're breaking down, and that causes physical weakness, right? Could lead to heart failure. Clumping of cells and clogging of small blood vessels. Ooh. Okay, so that's bad. That could lead to lots of problems. Okay, you can see here again, heart failure, brain damage, right? Because you have all those little tiny blood vessels in there and capillaries. Your spleen, look out. Your spleen's responsible for cleaning up your blood, and now you're throwing these little daggers into it all day. That's no good. And because of these things that are happening here, all the damage that this can cause, well, impaired mental function, paralysis, pneumonia, some other infections, rheumatism, right? It's when you're kind of attacking your own joints there, and kidney failure. So as you can see, this is why having sickle cell is so bad. It just ravages your system. Okay, now, yes, we did talk about carrying a copy of it might not be a terrible thing, right? Especially if you live around that Anopheles mosquito to avoid getting malaria, perhaps. But, oof, sickle cell itself, pleiotropy, right? One gene does a whole lot of bad stuff. And it's just yet another genetic curveball, right? Mendel probably wouldn't have been ready for something like this, where one little gene that he studied could have so many effects, right? Look at all the phenotypic effects from this one mutation that happens. Okay. So pleiotropy is when one gene has so many phenotypic effects, right? Well, what if you had more than one gene that does one phenotypic effect? That would be the exact opposite, right? And generally in Bio 1, we call that polygenic inheritance. Okay, and there's two examples I want to give you here about it. And we'll come back to like eye color in just a second. But the first one is talking about puppy dogs. So Labrador retrievers. Here you can see we have chocolate lab, we have a yellow lab, and we have a black lab. Did you ever know that these were based on epistasis genetic patterns? Here's how it happens. You have two genes for coat color in these dogs. You have the black or the brown alleles, right? That's it. Black or brown. And now you might be thinking to yourself, thinking ahead, oh, maybe this is incomplete, right? There's a blend or something. This is a little different because there's two genes involved. So what it is if you give black at all, big B, let's say, you're going to be black. You're going to be a black lab. If you give no big B's and only little b's, you're brown. So how in the world do you get the yellow lab then? There's a completely other gene that says, you know what? Go ahead and put pigment in or don't put pigment in. That's how it works. So you were either black or brown, but then there's another gene that says yes or no to pigment. And if that other gene is turned off, it doesn't matter if you're big B, big B, little b, or little b, you're going to be yellow. You're not going to have that pigment put into that fur. How cool is that? That's epistasis. That's when multiple genes are working together to give you a phenotype such as coat color. Okay? And there's another example here. Oh, there you can see it. Sorry there. Okay, so one gene determines the pigment color. Okay, and here you can see all the different dog types. All right, and sorry, get out of your way there. There you can see these beautiful animals, right? And here you can see, hey, this is like our 9:3:1 ratio, right? You have big little times big little. You should get a 9:3:1, but look what happens here. Anytime you have big B anything, you're going to be black. Anytime you don't have big B, you're going to be brown, as long as you have big E. And if you don't have the other gene that basically says, are you going to put pigment inside the hair? If you don't have the E gene dominant, then even if you're big B, or big B, or a little b, you're going to be yellow, right? No pigment. That's epistasis, right? So again, I go back to, could you imagine being Gregor Mendel if he, not only did he only look at one gene with two alleles and everything was fine, imagine if he looked at multiple genes coming together and these genes work together to form a phenotype. That would have been really hard, right, way back in the 1800s. So good thing that he didn't have to worry about multiple genes or epistasis. Let's take a look at another example here. Oops, sorry, polygenic inheritance. Now, the way this is described is a little goofy. Quantitative characters are those that vary in a population along a continuum. Quantitative variation usually indicates polygenic inheritance as an additive effect of two or more genes on a phenotype. For example, skin color. And this was the same way I showed eyes before. Eye color is another example. Now, this is confusing. So let's take a look at the next slide and figure out what exactly is happening here. Okay. So as you can see here, skin color exists along a continuum. Okay? Now, it's probably more than this, but what this is showing you is what if skin color for humans was only dictated by 3 genes? all right? And don't forget, pigment is usually dominant, right? So the more pigment, the more dominant the alleles are. So for example, what if you were big A, big A, big B, big B, big C, big C? Wow, you have like some super dark skin right here. Okay, darkest skin possible with just these 3 genes, right? And now what if you had little a, little b, little c, little c? You're close to an albino, right? You have super, super pale light skin, okay? And that's not most people on planet Earth. Most of us are in here somewhere, right? If you have light skin, you're probably in this range. If you have dark skin, you're probably in this range. A lot of us are right in between. We have like a tannish skin, okay? So that is called polygenic inheritance. It's quantitative. How many dominants did you get? And the amount of dominance you get gives you your skin color. Okay, so for example, let's just pick a person here, because the question always comes up, can 2 dark-skinned people have a light-skinned baby, or can 2 light-skinned people have a dark-skinned baby? So if we're in class together, that almost always comes up in class. And yeah, sure, let's take a look here. So let's pretend, okay, like for example, maybe this is me right here. Okay, I'm pretty pasty guy. So let's say that that's me. Okay, so for gene A, let's say, or the first gene, right, I have little a, big A. And then I'm little b, little c, little c. And let's say my wife, let's just say she's right here. Okay, so she's little a, little b, big C, little c. Right? So if you crossed myself and my wife, it would be, well, I have a 50% chance of giving a big A. And maybe I do. Let's say I gave a big A through meiosis, one of my sperm cells. Little b, I have no choice, and little c, I have no choice. So my sperm, let's just say, is big A, little b, little c. Now my wife has no choice for A or B. She's little a, little b, but maybe in an egg she put a big C in there. Now what if that specific sperm and that specific egg met? Yeah, we could have an offspring that's big A, little b, big C. Okay? And in that case, oh, you're a complete shade of skin darker. Right? Because now you have 2. It's quantitative. It's the amount of dominant that you have that determines, or the lack thereof of dominant, that determines your skin color. That's polygenic. And I'll give you a hint. If you're trying to figure out traits that are possibly caused by more than one gene, just take a look at humans, look at them very closely and say, is there a lot of variation? If there's a whole lot of variation, then you're looking at a polygenic trait. For example, my thumb, right? Hitchhiker's thumb. You can either do that and bend it backwards, or you can't. That's one gene, right? Being able to do this with your tongue, right? Make a U out of it if you can. Some of you can do that clover thing, which is really cool, but that's one gene. Okay, sometimes hair on a certain part of your digits here. So I have like hobbit hair on my hand. It's pretty cool, actually. But anyway, so there are genes, right? Single genes that do things, but a lot of times it's polygenic. It's the additive effect. So let's think of things that aren't either or in the population. Skin color, eye color, hair color, how tall you are, right? All of that stuff is very different from person to person, right? So those are all more than likely polygenic, okay? Now, if you have this polygenic inheritance there and you have all this variation, that's wonderful, because look at the phenotypic range that you get. You get a huge range of phenotypes. Just think if we're all sitting in class right now. How many of you are 5 foot or less? Not very many. A couple of you maybe, right? A couple little ones. And how many of you are over 6'2", right? Maybe 1 or 2 at the most, right? But most of us are somewhere in between. How many of you are over 5'4" but under 5'10"? Oh, there's going to be a huge number of that, and that's the bell-shaped curve. So when you're talking about polygenic inheritance, it's usually that nice bell-shaped curve that you get, okay? And that's just showing a lot of variation in the population. Okay, pretty cool, pretty cool. One last thing I want to talk to you about. Is genetics everything for your phenotypes? Of course not, right? We have nature and nurture. So there's a lot more than just your sequence of nucleotides that you got from your mom and dad shuffling the genes together, right, to make you. The environment plays a huge role, okay? And this argument is made all the time, and people are always saying, well, how much is nurture involved with who we are versus nature, like the sequence of our DNA, right? And they're both very important, especially as we learn more and more about epigenetics, which you'll get to when you take more bio classes. And all this is really saying is that, of course, the environment does play a role, right? So, for example, hey, you don't exercise enough and you sit and you pound chips like a champion. Well, guess what? You're going to carry around more weight than somebody else. Or what if your parents, made you watch certain YouTube videos growing up about ABCs and how to read and letters, and then they read books to you every night and they actually gave you a hug and things like that. Yeah, you're going to be very different educationally and just comfortably than somebody who had to sit in the corner all the time or was abused or something like that as a child, sadly. So your nature and nurture are both a big deal. Okay, here you can see some hydrangeas, and based on the soil acidity, right, the pH of the soil, you can have the same gene and it changes what they look like. Okay, the same alleles even, all because of the environment, right? Or me, like I said, I have some pretty pasty skin here, but if I go to the beach and I don't have enough sunscreen on, well, yeah, I'm going to look a whole shade darker. Maybe not darker, maybe redder. But point is, the environment does play a big role in your genes. Okay? So anyway, that's a little bit about curveballs. Okay? So I told you, Gregor Mendel, he got off lucky as far as he figured it out. He did a lot of really important, cool work, and he basically cracked the code of genetics. But now what we saw today was, there's a lot of twists and rule breakers and curveballs coming in where Mendel's rules are kind of like, they're twisted a little bit. Okay, they're not broken, it's just a little different. Okay, and that leaves us with one last thing to learn about, one last main thing, and that's going to be another curveball where your chromosomes matter. Okay, and don't forget, these genes are on chromosomes, so now we're going to have to think about that soon too. But that's in the future. So anyway, I hope this video was helpful, and good luck as you continue studying genetics. See you later.